In welcoming our next speaker, Sendurai Mani. Mani is ever looking young. Mani graduated from our department in year 2000 with working with Professor G. Padmanabhan, and he moved to MIT to work with Professor Robert Weinberg in the field of cancer biology. And year 2007, Mani moved southwards to Texas to start his own lab at the MD Anderson Cancer Center in Houston. And very recently, he was elevated to the rank of full professor in the Department of Translational Molecular Pathology. Mani's research program is mainly focused on understanding the signaling pathways during epithelial mesenchymal transition in a set of cells contexts in cultured human cells, patient-derived xenografts, and transgenic mouse models. And his lab is also looking for some cancer therapeutics. Um, so that said, great pleasure in welcoming Mani to request him to make a presentation of his work and give us some insights into cellular plasticity and its significance in cancer metastasis. Mani,
Thank
it's
you.
all yours.
Thank you. Can you hear me?
Yeah,
Wonderful. Thank you. Thank you, Dr. Maniappa. And, it gives me great pleasure and honor to give a lecture in this Sentinel symposium. And in 1993, when I walked into the biochemistry department, it was like a dream come true. So I came for an interview on the previous day night. So I didn't see the department. And the next day when I came for an interview where Dr. Maniappa, Dr. D.N. Rao, Dr. Naraja, and almost everyone was there in the interview. It was like, this was scary, but it was wonderful at the same time. They made you comfortable, and it was really more of a discussion rather than do you remember or do you understand? That was the thing which they were trying to understand. They were trying to find out. If you understand, they're fine. If you remember, that's not what they were looking for. Very nice. That really gave me a different perspective towards science, how we should think science. And then I got I was fortunate to do my PhD with Professor Padmanabhan. He gave a lot of freedom. So at the same time, so we got Dr. Rangarajan also joining the department as a faculty. So he was there in-house teaching us how to culture bacteria, do a column purification, whatnot. All the techniques I was fortunate to learn from Dr. Rangarajan. So this is a picture. I just have some picture I thought I would show. This is a picture. For whatever reason, my colleagues at that time, they were trying to push me into Professor GP's chair. I have no idea why. We were all having that fun at that time. And we used to work very late. And here is a lab, the old building. And here is a picture from the group. It was a great time. I thank and I really treasure every moment I spent on that campus. And here is a place where I came from Madurai. So in Bangalore, it was very cold on those days. So I used to wait for sun to come out. So you can just go out from the lab to stand out. And then, here is a place where all of us were just spending time. So in 1999, I moved to Bob Weinberg's lab. And that's where, it's Bob Weinberg. I started, I developed interest in understanding the biology of cancer metastasis. I will explain to you why and how, and then the significance and importance of this. So if you look at a cancer metastasis, it's quite a complex process where you have a primary tumor, and then the tumor become highly vascularized or become angiogenic, and then once the tumor cells at some point start leaving the primary site, getting into the vasculature through this intravasation. Now, the cells leaving the primary site travel to a distant site, they extravasate at a distant site, and they remain dormant for years, many years. At some point, they choose to proliferate and then create a new tumor or metastasis. Now the metastasis could reseed and create more secondary metastasis, so it goes on. So why do we care? The reason is, in this slide it shows first come first saved. For example, if somebody has a localized disease, independent of the disease subtype, cancer subtype, they tend to do better. For example, in prostate cancer, you can see the survival rate is extremely high if they have been diagnosed early when the tumor is localized. But there is a metastasis, then the survival rate is extremely low. So that's true for breast cancer, that's true for many cancers. So in India, you can notice that 7 in 10 patients, this is from India Today, patients walk to the doctor only at the terminal stage, which means they would have developed metastasis. So it's very important for us to understand the biology so that we can treat metastasis. At this point, we don't have many option to treat metastasis, but we can treat primary tumor very well. So the reason why, again, I got interested in this area is because when I joined Bob Weinberg lab in 1999, these two guys, they were taking Bill Hahn and Brian Ellenboss, they isolated normal human mammary epithelial cells. To that, they introduced SV40 early region telomerase and V12HRAS. This is SV40 inactivates large T and inactivates RB and P53, and the telomerase gives the continuous proliferative potential, and the HRAS kind of transforms them. When they did that, one of the very important observation they kind of noted was they were able to transform them by, when you inject them into the mouse mammary fat pad or mouse breast, they were able to form a tumor, but they were never metastatic. So that's when I got interested in saying, how can I make these tumors metastatic by introducing known set of genetic elements? So, so to cut the long story short, so if you look at a normal tissue, any breast tissue or any normal tissue, they are all beautifully organized, and they continue to express these various cell adhesion molecules such as tight junction proteins, gap junction, desmosome, etc., etc. This is what holds them together in one place. When a cell like this get transformed into a cancerous, they continue to express all these cell adhesion molecules. As a result, they remain local. So we hypothesized that a transformation will lead into generation of non-metastatic tumor, but in order for these cells to migrate from point A to point B, they need to break cell adhesion, and also they need to break cell matrix interaction. And we tested and proved in number of papers we published showing that this is what is happening during metastatic process. So I'm not going to go into that for the interest of time. So in addition, one of the another puzzle is that a pathologist, when they look at a tumor, let's say in a lung, and they could predict whether that particular tumor is coming from breast, prostate, or lung, or liver, or spleen, just by looking at a histopathology. And what we found previously is that if you take an epithelial cell and they undergo this EMT, become mesenchymal, and that's what lead to development of metastasis. So therefore, I hypothesized that cancer cells not only become mesenchymal following EMT, rather they become stem cells so that they could go and recreate a tumor histopathologically similar to the tumor where they come from. And in fact, that turned out to be true, and we published this story in this paper, which is one of the highly cited paper, which kind of shows that this paper made a huge impact beyond my lab. So this brings us to the next important problem, which is today, if you look at a tumor is made of both differentiated cancer cells and cancer stem cells, and the treatment what we have, or the conventional treatment, which predominantly targets differentiated cancer cells. As a result, you can reduce the tumor bulk, but then the cancer stem cells are not eliminated, and then they eventually proliferate and differentiate and create either tumor relapse or metastasis. And if you have a cancer stem cell specific targeted therapy, then you would be eliminating cancer stem cells. If you, in my, what we found, that even a differentiated cancer cells can de-differentiate through this EMT program, and then they can proliferate, eventually differentiate and create a tumor relapse. In other words, targeting cancer stem cells alone is not sufficient. So you need to combine both. You need to target both cancer stem cells and differentiated cancer cells. So our finding suggested that one could target EMT to eliminate cancer stem cells, and these are the things we are doing in the lab now, combining EMT specific inhibitors and the differentiated cancer cell inhibitors, and then see whether we can eliminate tumor. So I'm not going to discuss that part of the story. Rather, I'm just going to give you a paper which we just got accepted. This is showing that CD8 T cells inhibit metastasis, and then CXCL4 regulates its function. This story was led by Rubia Joseph and collaborated with various folks shown in this picture. Sorry, my slide is it's a big file. So similar to the story I was mentioning earlier, not only tumor cell can change its behavior from non-metastatic to metastatic, even the immune microenvironment can change from tumor killing immune microenvironment to immune suppressive and tumor promoting microenvironment. So you have various immune cells shown here. But the way we systematically approach this problem is by taking a two tumor line, one is called a 67NR, and one is called 41. They both are syngeneic tumor line. In other words, you can inject these tumor cells back into the mouse with an intact immune system as long as you are injecting them into the same background. These are biopsy derived cell lines, so you have to inject them into the biopsy mice. When you do that into the mouse breast, both develop tumor with the equal rate, which is shown in this panel B. But if you look for metastasis, these are luciferase labeled, so you can look for a metastasis to the lung. But again, we are looking for a metastasis from the mammary fat pad to the lung. When you do that, you see only 41 goes to the lung, but not 67NR. And you can also look for circulating tumor cells. You see only 41, the highly metastatic one, has metastasis or CTCs in the blood. So now, if you take the primary tumor of the two tumors 67NR 41 and look for CD31, which marks angiogenesis, there is no difference. If you look for a proliferation, there is no difference. But then when Rubia put these cells in suspension culture, asking can they survive in suspension, it's kind of mimicking what happens in the blood. They were able to survive equally, both of them. This suggested that maybe if we introduce these cells directly to the vasculature into the blood, they may be able to survive and develop metastasis. In fact, that's what she did. She injected, again in a wild type mice, she introduced 67NR and 41 through the tail vein. Now she was able to see metastasis to the lung, suggesting that here is a entire lung loaded with the tumors, both in 67NR and 41, suggesting that what they were missing, particularly the 67NR, was ability to get into the blood. If they manage to get into the blood, then they can complete the rest of the step. So what is the thing which is controlling the entry of 67NR going from primary site to the blood? So we thought instead of looking at a tumor cell point of view, we will look at it from an immune microenvironment point of view. So for that, she took not-skin mice, which you all know don't have a T cells, B cells, and NK cells. So introduced both 67NR and 41 into the mouse breast. And to our surprise, in this mouse, they found a tumor with a similar rate, the primary tumor. But if you look for metastasis, even 67NR was able to develop metastasis. And you can see there is no difference in number of CTCs. This suggested to us that what was missing or what was stopping from 67NR developing metastasis is the immune cells, but it could be T cells, B cells, or NK cells. So then we have a biopsy nude mice. So we bought a biopsy nude mice, injected both the cells into the mouse mammary fat pad, and you could see again they formed a tumor with equal rate. And if you look for a metastasis to the lung, we were able to detect metastasis even in this case by 67NR. the nude mice, they don't have a T cells. So now we have narrowed down to T cells and eliminated the other two cell types, B cells and NK cells. Now within T cells, as you all know, you have many subtypes. So we collaborated with Sean Zhang, and he backcrossed CD8 knockout mice into the biopsy background, and then in that Rubia injected either into the wild type or into the CD8 knockout mice. For some reason, the tumor grew pretty big in the absence of CD8 T cells. And when we look for metastasis, in this case, she's putting only 67NR. In the absence of CD8, the 67NR cells, otherwise non-metastatic, started developing metastasis or migrating from mammary fat pad to the lung. So this kind of, we also tested CD4. It is not CD4, and maybe I don't have that. So it's not CD4, and we reconfirmed that this finding using function blocking antibody as well. So now we want to know how CD8 is regulating metastasis. So when she took cells 67NR41, either grown in a petri dish or grown in a wild type mice or in a nude mice, and she performed a growth factor or cytokine array, what she found is that the expected kind of cytokine was something which should not be expressed when these cells are cultured in a petri dish, and it should not be expressed in a cell 67NR when it is injected to the wild type, but it should be expressed in 67NR when it's put in a nude mice. It should also be expressed in 41 in a wild type as well as in nude mice. So we scanned number of cytokines. The only one fit our hypothesis, our expectation, is CXCL4. I will explain to you what is CXCL4. CXCL4 is it's a cytokine predominantly secreted by platelet. It's also called a PF4, platelet-derived factor 4. What Rubia did, she bought a CXCL4 from a company. She administered mice having a 67NR tumor with CXCL4. And what she found, even just by administering CXCL4 in a wild type mice, she was able to make a non-metastatic 67NR tumor to become metastatic. And you could see more CTC, suggesting that CXCL4 is one of the rate limiting factor for these cells to develop metastasis. So another thing we wanted to know is how CXCL4 interacts with the CD8 T cells, right? That's something. So the CXCL4 is known to act through CXCR3. So she went ahead and blocked CXCR3 in a highly metastatic tumor, which is the 41, and she was able to reduce the metastasis of those cells as well in a 41 in a wild type mice. So now the question is, who is producing CXCL4? We know it is previously it is known that it is platelets, and we reconfirmed it. We grow the cells 67NR and 41 in a petri dish. They don't produce any CXCL4. These cells are labeled with RFP, so we put them in a biopsy wild type mice and then isolate RFP positive cells of 67NR41. They don't produce any CXCL4. We also isolated CD45 positive fraction, which is an immune cells, and they also don't express any CXCL4. The only platelets, if we isolated from these mouse tumor, they produce CXCL4, and you can see we went ahead and checked their presence in 67NR41, and you can see the CD8 is highly abundant in 67NR, but in 41 they are less. But if you look for a CD61, which marks platelets, they are low in 67NR and slightly high in 41, and that's quantified here. In fact, we did something called IMC site-off, where you can label nearly 30 antibodies with the different metal, and then you can label cells in these tumors and look for presence of these various cell types. And what you see here, particularly CD8 cells, 67NR has more CD8 T cells compared to 41. So we went ahead and also tested the same idea in a human tumor tissue microarray here in collaboration with Dr. Fei Yang. So you can see patients with CD8 positive cells tend to have a low CD61, and the patients who had a low CD8, they had more CD61. and which is quantified. Most importantly, the patients who had high CD8 and low platelet, they did better compared to the patients who had a low CD8 and high platelets. So overall, this kind of told us that the tumor cells, they are, they trying to develop metastasis, but CD8 T cells puts a break on them, and platelets by somehow promote the metastasis, which is kind of known in the literature, the right part, not the left part. So then we were, Ruby, I was interested in understanding how platelet is able to manipulate CD8 T cells, or how the CD8 T cells manipulate platelets. So what she found is that when you take CD8 T cells, incubate with platelets, for some reason platelets turn on annexin 5, suggesting that they may be dying. But the issue is they don't have nucleus, so we didn't pursue that further. Rather, we asked, can platelet manipulate or modulate the function of CD8 T cells? So she isolated platelets or the CXCL4 and then incubated them with the monocytes. The reason is we do know that MDSCs are another cell type. So just one step back, when you take a platelets or CXCL4, incubate them with the CD8 T cells, she observed nothing. The platelets or CXCL4 on their own not able to impact the function of CD8 T cells. So therefore, we hypothesized that it must be acting indirectly through a cell type such as MDSC. So she took platelets and CXCL4, incubated with the monocytes, and asked, can they make more MDSC? In fact, we were able to make more MDSC. And those MDSCs were able to go and inhibit the function of CD8 T cells. I'm not showing you all the data for the interest of time, but we could demonstrate that platelets as well as CXCL4 produce more MDSC from monocytes, and those MDSCs inhibit CD8 T cells, and that's how it works. So here is another data where we inhibit MDSC function, which is myeloid-derived suppressor cells. You could see in a 41, the highly metastatic tumor, the metastasis is inhibited. The most important finding is there's another postdoc, his name is Suhas Vasekar. He used to be in my lab. He took entire TCGA data set and he analyzed the correlation of MDSC with platelets, and you could see that whenever you have a platelets, they tend to have an MDSC across various cancer types, not just in breast cancer. If you look at a CD8 and MDSC, which is known in the literature that they are negatively correlated. But this is another thing which we found, that if you take a CD8 and platelets, they are also negatively correlated across various cancer types, not just in breast cancer. And they also predict, this various combination predict poor clinical outcome. For example, here is a survival probability where you have a CD8 low, platelet high, MDSC high is bad for cancer patients compared to CD8 high, platelet low, MDSC low. So in summary, this part is that the tumor cells in general, the CD8 T cells inhibit tumor cell migration, invasion, and metastasis, and CD8 T cells also inhibit platelets. So as a result, you have a high CD8 and low platelets. Under that condition, you tend to have no metastasis. But at some point, we don't know how and what that triggers, that platelets find a way to survive. Once they overtake it, now they are able to promote the migration, invasion of tumor cells. Simultaneously, they make more MDSC, which inhibits CD8 T cells. Collectively, this leads to metastasis. So that's a summary of the paper. Finally, in the last 2 minutes or 3 minutes, I just want to present you with a new initiative which we started. Probably you all know precision medicine, particularly the undergrads and the grad students, where if you think about patients with the treatment, like most of the treatment works for only one or few patients. For rest of them, it does nothing, particularly the cancer treatment. So here is the data, like, the conventional treatment is you take cancer patients, you give a standard treatment. So some patients benefit from that, but not everyone, right? For some patients, there's no effect at all, as is shown in this graph. But for many patients, they also have a side effect. So which is a common problem today. So the precision treatment, where you cancer patients, you profile them based on various genomic profiling. Then you separate them into group A, B, and C, and give their respective treatment, right? If they have a mutation in a particular gene and you give their treatment, if they have a mutation in particular condition B and you give their treatment, and now you achieve effective treatment. So this is a classic example where a melanoma patient with the BRAF mutation was given with the vemurafenib, and you could see a patient with so many tumors all over. The patient recovered significantly in panel B, you could see, but the tumor relapsed, right? That's a problem because the tumor is constantly evolving unless otherwise you eliminate every single tumor cell, which is difficult. But at least this patient had another 23 weeks of extra survival, and hopefully they find another mutation and you could go and target. So today it's kind of where we are is that integrating various omics data and then putting them all together and treating them. So that's kind of a future. So the way I see is that you have a various omics, feed them into supercomputer that can go into the phone, and that will create cocktail of less toxic medicine. The patients recover. So unfortunately, this is far away from today. We will get there, but not today. So what we did, we created a small consultation company where these guys plus many others, they all joined as an advisors, and this is what they do for their living. They look at these profiles and then they predict medicine. So fortunately they all agreed to work with us and through the phone, and we can use their expertise to design these less toxic medicine. So that's, we just formed a company called IISc Precision Oncology. It's kind of very moderate fee, just more like a service. This Dr. Seventhi Limaye and we started this company called IISc, which is for a precision medicine. She's the one who treated Sanjay Dutt, for example, whom now considered cancer free almost because she applied precision oncology. So this is another thing which we are almost there. Probably in a month we will be out there, but I just want to put this idea out that particularly in an Indian context, this would be very beneficial for patients with advanced cancer. So in summary, I've shown that EMT program play a central role in regulating metastasis. EMT program induces stem cell properties on a differentiated epithelial cells. The CD8 T cells regulate metastasis in a platelet-dependent manner. So these are the people whom I collaborated. I didn't get a chance to speak a lot of work by these folks. So this is my group. So this is a funding support, and I thank you all for your attention. Thank you.
Thanks, Mani. And any questions, comments for Mani?
Dr. Mani, this is Suraj Bhatt from UCLA. How are you doing?
Good. How are you?
That was a terrific talk, and so was Dr. Subramaniam's talk before that. That was a terrific talk. I was just wondering that since 80 to 90% of all cancers come from epithelial cells, how critical is the EMT? Does EMT happen before metastasis or after metastasis? Is it critical?
It's extremely critical. So originally when we hypothesized that EMT is critical, we were trying to understand that whether cell need to undergo complete mesenchymal transition or can they become mesenchymal, just acquire some mesenchymal properties. So what we found today, or what we know today, is that the cancer cell need not become mesenchymal. All they have to do is they have to inherit some mesenchymal properties. So that kind of, when they inherit some mesenchymal property, they also tend to get some stem cell properties. Together, that helps them to leave the primary site, manage to survive in the blood. The key is ability to survive, and then establish a tumor at a distant site. So it's EMT is critical, not like the way we originally thought in 2000. You need to become mesenchymal, but you don't have to lose epithelial.
So if I may, can I ask a follow-up on that?
Of course you can, yeah.
CD8 knockout cells, what is the proportion of stem cells, tumor stem cells, in those mice.
It's a good point. So that's something we are looking at right now. In other words, so originally we looked at it from a tumor cell point of view, right? How the tumor cell will manifest the immune microenvironment, not immune microenvironment, the metastasis. So then now we looked at how the immune cells manifest or manipulates the tumor cells. One of my postdoc, he took 15 different mouse lines, syngeneic line. We try to use mostly syngeneic line simply because that allows us to model immune cells as well. And then he's asking, what is the contribution of EMT in manifesting the immune cells? So he has a nice finding showing that number of immune cells, particularly the immune suppressive cells, they get turned on following activation of this EMT program. So we're trying to understand that. So
that's a terrific interface between stem cells and immune system. Thank you.
Yeah.
So can I ask a quick question, Dr. Muniappa?
Of course you can, Sukumar. It's good to see you.
Yes. So nice to see you again. I mean, a lot of you talk tomorrow. Sundar, a great talk as always. I'm always a big fan of your work, as you know. So nice to see you as well, and good morning. I had a very quick question. The CXCL4 data you showed, do you have any data or any information on how it regulates stemness or EMT per se, or does it have any effect on the stemness directly, or the effects you are seeing are totally CD8 mediated cytotoxicity, that effect? Is there any data available on CXCR4 expression on the stem-like cell? Does it change during EMT, anything like that? Yes, you are still mute. Yeah.
Somebody muted me. I didn't mute it. So, it's a good point. That's another thing we are kind of puzzled. Like, what triggers tumor cells to, or the platelet? When the platelet manage to survive in the tumor microenvironment, then they start producing the CXCL4. But how the CXCL4 impacts the tumor cells, we don't know that. We don't know. We didn't explore that. All we know is if you add a CXCL4 to a tumor cells, they tend to express CXCR3, the receptor. They express more, they become more highly migratory and invasive and all that. But we don't know its impact on stemness. It's a good point. Thank you.
Kumar? Yeah, Kumar, yes.
Manish, this is Kumar from MCB department. Please let me correct me if I'm wrong. What I understood from your talk, you mentioned 67 NR breast cancer cell line, which is not metastatic to begin with, became metastatic in a nude mice. Thereby, you identified CD8 cells are the one responsible for inhibiting their ability to metastasize. Okay. Now, earlier you have shown the ability of cancer cells to metastasize depends on their ability to undergo EMT. Okay. And which is also kind of overlapping with the stem cell phenotype, which you had discussed when you came to give a talk some time ago, a couple of years ago. Now, can you please reconcile this the ability to undergo EMT, stemness property, that makes them metastatic versus in today's finding that absence of CD8 T cells only made 67 NR metastatic. That is means 67 NR is not EMT cell line. Is that the reason originally it is not metastatic? And can you please combine these things and give your opinion on that? Thanks.
Absolutely. Thank you. That's what we are right now examining right now as you speak, as you just said. So again, in a tumor, you have epithelial, you have a mesenchymal. There are something in the middle. They kind of acquired mesenchymal, but not completely mesenchymal. So the 67NR is more of a mesenchymal. So when a tumor cell in the primary site itself, they undergo this extreme differentiation, they lose the ability to develop metastasis. But if they are extremely epithelial, then also they don't. Probably the immune cells tend to kind of inhibit them once they undergo the complete differentiation into mesenchymal program. So they need to have this dual personality where they could be epithelial or mesenchymal. So that's one of the area we are right now examining. So we have taken again several models, and if you take again syngeneic tumor lines with extreme mesenchymal, they are differentiated into mesenchymal. They lack the ability to differentiate back to epithelial. Under that scenario, they cannot develop metastasis even if they've undergone an EMT. Okay, so 67NR is one such example where the cells have undergone a complete mesenchymal transition. They're differentiated. There is another tumor model we use, something called a T11, which is developed by Jeff Rosen's lab here in Baylor. They've become a mesenchymal transition. They also don't develop metastasis from the mammary fat pad. But if you put them in the blood, inject them directly to the tail vein, they do develop metastasis. So you need to be in the middle, midway, not right or left.
Mani, we have a question in the chat box. This is from Iris Sivatson. I'll read out the question. What are the CSC numbers with respect to metastatic versus non-metastatic cells? And they get reduced with chemotherapy, but then they need to come back for normal cell functions. So your response.
Thank you. I think so initially we thought that it could be just a simply stem cell number, but then later we realized that it's not just a stem cell number, it's a stem cell quality. So there's a guy with the name Max Vicha who's at the University of Michigan. He found that the stem cells in a tumor, they tend to be less plastic. In other words, he isolated stem cells with a CD44 and something called ALDH, and he found that the stem cells which are more of a tumor stem cells are tumor initiating stem cell. If you remember, any tumor in order to grow, they need a stem cell, but those are tumor initiating stem cells. And there are stem cells which are capable of developing metastasis. They acquire something more than just a tumor initiating stem cells. So the tumor stem cell number may be different between different tumors, but the quality more than quantity of these stem cells are different between metastatic and non-metastatic tumors. And the chemotherapy definitely works, depending on the chemotherapy, works on both the they don't work on stem cells. So that's a problem. On top of that, chemotherapy can induce inflammation, and inflammation can cause EMT and make more stem cells. So most of the time when patients are treated with the chemotherapy, they tend to left with stem cells rather than differentiated cells. Hope that answers the question.
Mani, finally, I have another question from my colleague. This is Purusharath Rajaguru. Is it possible for tumor cells to release some factors to trigger platelets to produce CXCL4?
Good point. I think it's a very good point. We don't know what those factors are. So one of the things we've done is we just got a new machine instrument called a Rhapsody, BD Rhapsody by BD. So what it does is you can sequence single cell, both the immune cell as well as tumor cell. So we are sequencing as we speak all these 15 syngeneic line plus patient tumor tissue at a single cell level, hoping that would yield us that information what you just asked. And we are also taking all these tumors, performing something called a Luminex assay. What it does is that it looks for cytokines specifically produced by these tumors at a greater extent. So hopefully these two will help us identify those factors what you just mentioned, Dr. Munyappa. Thank you. Really.
Well, Mani, I think I should wrap up in the interest of time. So thanks again for telling us your very, very interesting work. I hope we'll have opportunity to meet you and talk to you more in detail sometime in the future. And more importantly, thanks for joining us with all your the weather conditions and pandemic and all that in Houston. All the best.
Thank you so much, and thanks everyone. It's a pleasure. Thank you. I really appreciate the opportunity.